Assalamualaikum and a very good morning, ladies and gentlemen. Salam Ramadan to our Muslim participants around the world. Welcome to the IMR-SEAMEO Webinar Series 2026, brought to you by the Institute for Medical Research IMR Malaysia, which also serves as SEAMEO TropMed Malaysia, a regional centre under the Southeast Asian Ministers of Education Organisation, SEAMEO. We are honoured to welcome participants from across Malaysia and beyond, including distinguished research committee members, academia, the healthcare sector and government agencies. I am Dr. Nurul Hidayah, Medical Officer from Bioassay Unit, Herbal Medicine Research Centre, IMR, and I'll be the moderator for this session. Thank you for taking the time to join us today. Before we start the presentation, allow me to share a few essential housekeeping notes from Secretariat. First, this session is streamed live on YouTube channel of NIH Malaysia. We will not be taking questions during the presentation. However, please submit your questions through the link shared in the YouTube chat as we will address them in the question and answer session after both presentations. An attendance link will be provided in the chat on YouTube. Please fill it out if you wish to receive an e-certificate. Last but not least, please subscribe to the NIH Malaysia YouTube channel and click the notification bell to stay updated on our latest posts. Now, ladies and gentlemen, our theme today is why drug candidates fail in clinical trials, the critical role of comprehensive preclinical evaluation. And for this team, we have two presenters who are experienced researchers from our very own Bioassay Unit Herbal Medicine Research Centre. A brief introduction on Bioassay Unit. Our unit conducts scientific studies to evaluate the efficacy and safety of local medicinal plants for herbal medicine development. Established in 2001, this unit specializes in vitro and in vivo research, including anti-infective, immunomodulatory, anti-aging, and bioavailability studies. Using cell-based assays and animal models, the team investigates biological activities, mechanisms of action, and pharmacokinetic profiles to generate strong preclinical data that support clinical studies and therapeutic claims. Without further delay, let me introduce our first speaker. Dr. Muhammad Ridzuan bin Muhammad Abdul Razak holds a Bachelor of Science and Master of Science in Biochemistry from University Kebangsaan Malaysia and earned his PhD in Structural and Molecular Biology from University College London in 2012. He joined the Institute for Medical Research IMR in 2004 and is currently the head of the Bioassay Unit at the Herbal Medicine Research Centre IMR under the Ministry of Health Malaysia. He has extensive experience in herbal medicine research, particularly in dengue, malaria, and COVID-19. Currently, he is a member of the Dengue Alliance Preclinical Working Group under Ministry of Health Malaysia, and he also serves as committee member of SEAMEO TropMed Regional Centre for Microbiology, Parasitology, and Entomology, and the Animal Care and Use Committee, Ministry of Health Malaysia. In addition, he is the lead QA officer for the GLP-certified non-clinical research facility at the Institute for Medical Research. Presenting on the topic, the power of preclinical studies in drug discovery, let's welcome Dr. Ridzuan. Screen is yours, Doctor.
Thank you for your kind introductions, Dr. Hidayah. First of all, I would like to thank the organizer for IMR-SEAMEO Webinar for giving me this opportunity to present and share some knowledge in this webinar. Assalamualaikum and good morning, everyone. Thank you for joining. I'm Dr. Muhammad Ridzuan, Head of Bioassay Unit, Herbal Medicine Research Centre, IMR. So today, I'm going to present about the power of preclinical studies, which is the most critical first step in drug development. Bringing a new drug to patient is a long journey because it takes about 10 to 15 years from lab bench to the bedside. So basically, scientists find the problem or target in the body. Then they search and design a chemical and extract the natural compounds that could help. Before testing in people, the preclinical studies were performed. If those goes well, the drug moves to human trials, get approved by authorities, and eventually reach patients. The truth is, most drugs that are tested in the lab never make it to patients. Only about 1 in 5,000 do. And as we look at the preclinical study phase, the number of drug candidates are actually reduced before entering the clinical trials. In this case, the preclinical studies are actually a gatekeeper that help to spot problems early, so we don't waste time, money, or to put people at risk. So what are preclinical studies? Before a new medicine can be used on people, scientists must first make sure it is safe and likely to work. This early testing is called a preclinical studies. Think of it as a safety check before moving to human testing. First, researchers test the medicine in the laboratory using cells. They look at simple questions. Does it work? Does it kill the virus or reduce inflammation? Is it harmful to normal cells? If the results look promising, the next step is testing in animals. This helps scientists understand, is it safe in the living body? What dose is safe? What does the body process the medicine? And does it actually improve the disease? Only after passing these tests, the drug candidate can move to human trials, and the data is required by regulatory authorities such as FDA. In simple words, preclinical studies are like a trial run to make sure a new treatment is safe and worth testing on people. It protects human volunteers, enrich patients, and prevent later in clinical trials. So before any new drugs reach patients, it must first prove itself in the lab and in animals. That is the role of preclinical research. Preclinical studies consist of 3 important experiments that need to be conducted. First is pharmacokinetics, second one is pharmacodynamics, and the last one is the toxicology experiment. On this slide, we are looking at pharmacokinetics or PK. Pharmacokinetics simply means what the body does to the drug. In this example, we give the drugs to mouse either by injection or oral gavage. After the drug enters the body, 4 main things happen. First is the absorption. This is how the drug enters the bloodstream. Second is the distribution. Once in the blood, the drug travels around the body and reaches different organs. Third is metabolism. The body, mainly the liver, breaks down the drug into smaller components. Finally, we have excretion. The drug or its breakdown product leave the body, usually through urine or feces. So, in simple terms, pharmacokinetics tracks the journey of the drug from how it enters, where it goes, how it is changed, and how it leaves. Understanding this helps us decide the right dose, how often to give the drug, and whether the drug stays long enough to be effective. Now let's move to pharmacodynamics, or PD. If pharmacokinetic is what the body does to the drugs, the pharmacodynamic is what the drug does to the body. After administration, the drug travels to target organ or specific receptor. This is very important because the drug must interact with the right biological targets to produce a therapeutic effect such as reduction in viral load, decrease inflammatory markers, lower fever, improve survival, and reduce tissue damage. This response is what we measure to determine if the drug is effective. If in preclinical studies, pharmacodynamics help us answer key questions. Does the drug work? How strong is the effect? What dose give the best response? Is the effect dose dependent? On this slide, we focus on preclinical toxicology studies, which help us answer the critical questions: is the drug safe before we move into human trials? Before we administer any compounds or humans, the safety of the compound must be evaluated thoroughly in animal models. First, the acute toxicity study is conducted. where a single high dose of the compound is administered. This is followed by monitoring of animal clinical signs, survival, and toxic reactions. This will give us an estimate of the lethal dose range. Next step will be subacute or chronic toxicity studies, which will be conducted depending on the type of disease or treatment duration. In toxicology studies, the genotoxicity will be conducted in order to see whether the compound damage DNA, because DNA damage may lead to mutation or cancer risk. The developmental and reproductive toxicity is also important to study whether the drug affects fertility, pregnancy, or fetal development. Last but not least, the safety pharmacology is also important to be conducted in order to see the effect of the drugs on the vital systems such as heart, brain, and lungs. Throughout all these studies, we measure key endpoints: mortality, organ toxicity, hematology and biochemical changes, and most importantly, we determine the no observed adverse effect level to calculate a safe starting dose for first-in-human trials. In summary, preclinical toxicology allows us to move into clinical development with confidence. Now let's look to something very critical in drug development. Pharmacokinetics, pharmacodynamics, and toxicology are closely interconnected. We cannot study them in isolations. In simple terms, PK drive exposure, PD drive efficacy, and toxicology defines safety limits. And together, they guide dose selection for clinical trials. That is why integrated PK, PD, and toxicology studies are essential before moving any compounds into human. Now let's try to answer these questions. Why clinical trials fail? This paper highlighted that the lack of robust preclinical experiments is one of the factors that causing the clinical trials, especially the phase 3, fail to meet the clinical outcome. Now let's try. Sorry, some technical error here. Many times, preclinical experiments are tested in a very narrow set of experimental conditions. So, when such approaches are finally tested in the context of human disease, the challenge provided by the varied age of patients, the complex genetic makeup of human populations, and the complexity of diseases to be treated provide challenges which were never tested or modeled. Therefore, the robustness of preclinical procedures should indicate its treatment efficacy in spite of variation in the age, size, and genetic composition of the disease models. The gap usually lies in two areas. First is lack of reproducibility. Second is poor translatability. Let me start with reproducibility. To make efficacy studies reproducible, we must first standardize our experimental design. This means clearly defining the animal strain, age, sex, infection, or disease induction protocol, and primary endpoints before the experiment begins. Everything should be written in a protocol, not decided after we see the results. Second, we must reduce bias. Randomization and blinding are essential. For example, animals should be randomly assigned into treatment groups, and outcome assessment, including histopathology scoring, should be blinded. We also need a proper sample size calculation to avoid underpowered studies. Third, we must validate our tools. authenticate cell lines, check for contamination, use validated cells, include positive controls, and ensure instruments are calibrated. In general, reproducibility depends on technical rigor. Now, reproducibility alone is not enough. We also need translatability, meaning the findings should reflect what happened in humans. To improve translatability, we must acknowledge that humans are biologically complex. Therefore, our preclinical model should reflect the complexity. Instead of testing only one strain of animal, we should consider different strains. Instead of using only young male animals, we should include both sexes and different age groups. Where relevant, we should also consider comorbid models. Next, we must also use clinical relevant endpoints. The outcomes we measure in animals should align with what clinicians measure in patients, such as viral load, inflammatory markers, organ function, or survival. Biomarker alignment increases the chance that preclinical findings will predict clinical outcomes. Dosing is another critical factor. We must ensure that dose used in animals reflect exposure levels that are realistically achievable in humans. Excess dose exposure may show efficacy in animals, but fail in clinical trials. Finally, demonstrating mechanism of action ideally confirmed in human cells may strengthen biological explanations and regulatory confidence. This is one example of success story of preclinical study of COVID-19 antiviral drug produced by Pfizer. This drug is currently marketed as Paxlovid and very effective against different variants of SARS-CoV-2. The drug is currently marketed as Paxlovid. The Paxlovid is actually made with a combination of nirmatrelvir, which is the antiviral agent, and the ritonavir, a cytochrome inactivator that helps to improve the bioavailability of nirmatrelvir. I'm not going to discuss in detail about this paper, just to highlight some of the important points that showed the robustness of the study. First is the in vitro antiviral study on nirmatrelvir, which is the antiviral drug, was tested against different cell lines, such as monkey kidney cells, human lung epithelial cells, and normal human bronchial epithelial cells. Second is the small mouse model in this study, which the mouse was actually intravenously infected with dengue mouse adapted COVID-19 virus to ensure the optimum infections and disease development in the model. Here we can see that the reduction of viral load in the target organ was observed after treatment with nirmatrelvir. Third, the study was also involved the bioavailability and toxicokinetic assessment. In this case, the bioavailability of the nirmatrelvir with and without ritonavir combination was studied in more than one animal species, such as rat and monkey. Of course, bioavailability data in humans has also strengthened the data of the study. So what we can learn from this study is that the experiment must be conducted in different models or in a model that is closest to human to ensure the robustness of the preclinical studies. Now let's look at the future of preclinical drug discovery. On December 29, 2022, President Biden signed the FDA Modernization Act 2.0 into law. This comprehensive legislation permits the utilization of specific alternatives to animal testing, including cell-based assays such as human-induced pluripotent stem cells, organoids, and organ-on-chips, and advanced artificial intelligence methods to predict drug toxicity, metabolism, and other ADME properties. This paper highlighted that one of the main factors that caused the phase 1 and 2 clinical trials failure is lack of genetic diversity, and pharmacogenomic differences between animals and humans. This paper also proposed an innovative concept called cell village. This alternative technique cultivates multiple iPSC cell lines together and differentiated into various cell types and incorporated into 3D models such as organoids and organ-on-chip, which is supposed to closely mimicking the human in vivo environment. These cell village tissues can then be subjected to therapeutic agents, and each cell line's gene expression response will be detected by the single-cell technologies. This transformative approach is potential for enhancing our understanding of drug safety and efficacy, ultimately increasing the likelihood of therapeutic success in early clinical trials. However, at the moment, this technique is costly, not high throughput, and still under development process to actually mimicking the genetic diversity of human population. As a conclusion, preclinical experiments are important as a gatekeeper in the drug discovery and development pathway to ensure that the effective and safe candidates enter clinical trials. Standardizations of the experimental techniques or assays build reproducibility. Inclusion of biological diversity factors in the study will improve translatability. Both reproducibility and translatability are essential to reduce clinical trial. With that, I end my presentations. Here is my contact information for further discussion, clarification, and potential collaboration. Thank you for your attention. Over to you, Dr. Hidayah.
Dr. Ridzuan for the very informative presentation, emphasizing the vital role of strong and well-designed preclinical studies in ensuring drug safety, effectiveness, and greater success in clinical trials. So if you have any questions for Dr. Ridzuan on his presentation, kindly submit using the link on the YouTube channel. Moving on, allow me to introduce our next speaker. Dr. Norzahirah Ahmad recently completed her doctoral research where she studied methods for real-time measurement of pharmacokinetics. Her research interests focus on pharmacokinetics, herbal medicine, and understanding how drugs and natural compounds behave in the body. Her work helps determine appropriate dose selection to ensure both safety and therapeutic effectiveness. With the topic pharmacokinetic, make or break factor in drug development, passing the screen to you, Dr. Zahira.
Thank you very much, Dr. Hidayah, for the kind introduction. I'm Norzahirah, and I'll be introducing the concept of pharmacokinetics and why it is considered a make or break factor in drug development. Where does something goes in the body after we take it? Whether it is medicine or tablet or capsules or other examples such as energy drinks that contains caffeine or traditional preparation like turmeric juice, what happens to them after they enter our body? That journey from entry to exit is called pharmacokinetics. In this presentation, I will focus on what happens when we take medicine by mouth or orally. After we swallow the medicine, it goes to the stomach where it is dissolved in the stomach acids. The soluble medicine then moves into the small intestine and then to the portal vein where absorption starts to happen. The medicine then travels to the liver, which is the site for metabolism. Liver enzymes then chemically transform the drug into another form. Notice the blue color change here. After that, the transformed medicine then enters systemic circulation where it is pumped by the heart throughout the body to reach the target location. Over time, the medicine is eliminated mainly through the kidneys and excreted in the urine. Scientifically, pharmacokinetics studies the changes to the amount of medicine in the body as it goes through the processes of absorption, metabolism, distribution, or elimination, which is also collectively known as ADME or ADME. Successful drug development, excuse me. Successful drug development requires careful consideration of ADME factors. First, let's talk about absorption. Traveling through the stomach and then the small intestine can change the drug behavior. This is because the different parts of the gut have different pH levels. pH level simply means the level of acidity. Being in a fat state, for example, after eating, or being in a fasted state or empty stomach will also affect the pH levels. This then affects how the drug ionizes and how it solubilizes and also how stable it is. Another factor to consider at this stage is membrane permeability. Remember, for a drug to work, it must enter the blood. The crossover to the blood is necessary, and because our cell walls are mostly made of fats, drugs that can mix well with fats are able to pass through more easily. This is called lipophilicity. Therefore, for good absorption, three things matter. It must dissolve well, stay stable in the different pH conditions, and be able to pass through the intestinal wall. Next, we need to consider what happens in the gut and liver in terms of metabolism. Now, in the gut, there is something called the transporters. We can think of them like pumps. These transporters can pump the drug back out, preventing it from entering the blood. Meanwhile, in the liver, there are enzymes that can break down the drug and makes the drug ineffective. So, for a drug to work well, it should not be broken down too quickly and it should not be pumped out by the transporters. After overcoming the challenges of the stomach, the small intestine, the liver, the drug finally makes it to the blood to be circulated. But the blood contains plasma proteins. These plasma proteins bind to the drug, reducing the number of free drugs that can travel to the target site. Additionally, there are also physical barriers to the target site, such as the blood-brain barrier, if the drug is trying to reach the brain. Or, in the case of cancer, the tumor microenvironment can act as a barrier. The tumor has its own unique environment with abnormal blood vessels and also dense tissues, which can prevent the drug from reaching the cancer cells effectively. Finally, the drug must be eliminated from the body, usually through the kidneys. Drugs that are hydrophilic, which means they are water-loving, are generally removed more easily in the urine. Drugs that are lipophilic, meaning fat-loving, tend to stay longer in the body because they can hide in fat tissues, and if they stay too long or accumulate too much, they may cause toxicity. So, in distribution and elimination, attention is to be paid to the percentage of the protein binding, appropriate lipophilicity, and also the potential toxicity of the drug. Now, let's talk about the oral pharmacokinetics during the preclinical stage in animal models, in this case, the rat. First, drug is given to the animals, and this is called the dosing step. Then, blood samples are collected at different time points, for example, 1, 3, 6, 12, 24, or 48 hours. These blood samples are then analyzed using LC-MS to measure how much drug is present in the blood samples at each time point. We can then plot the drug concentration against time, and this is the typical curve. For oral dosing, the concentration rises first, and this is the absorption phase. It then peaks and then slowly declines as the drug is distributed and metabolized and also eliminated. If the drug is given intravenously, which means directly to the vein, the concentration starts high and declines over time. From these two curves, we can then apply mathematical modeling to calculate the important PK parameters. These parameters tells us about the systemic exposure of the drug and how much the drug is exposed to in the body over time. As we have now learned, blood is the main site for drug monitoring. The limitation of this approach is that we only have snapshots, like taking photos at 1 hour and 3 hours, but we do not know what is happening between the intervals. In this recent work conducted at Osaka University, instead of blood, we are now looking at the skin to measure drug concentration. At the same time, we monitor the ECG to observe the drug's effect on the body. This allowed us to measure both the PK, which is how the drug concentration changes over time, and also the PD, which is how the drug affects the body continuously and in real time. This study represents the basis of the development of a wearable sensor prototype, moving from intermittent blood sampling to continuous and real-time monitoring. Now, let's talk about pharmacokinetics for complex mixtures. Unlike a single drug, herbal formulations contain multiple active compounds. This means each compound can behave differently in the body. In this graph, each colored line represents a different compound. They are absorbed at the different rates and reach different peaks and are also eliminated at different times. Hence, PK measurement becomes more complex as every active ingredient must be measured individually. Despite this challenge, understanding this helps us to know which compounds contribute to the overall effect as well as their effective timing. We can now see that no matter how promising a drug is in the lab, if it cannot be absorbed properly, reach the target, remain stable enough, and be safely eliminated, it simply will not work in real life. The take-home message for the researchers listening is simple. Efficacy alone is not enough. Pharmacokinetics consideration must be given as early as possible. And finally, pharmacokinetics is not just about drugs in the lab as it applies to everyday life. When we drink coffee, take medicine, or consume herbal preparations, our body processes them in the same way. They are absorbed, distributed, metabolized, and eliminated. For example, caffeine from coffee is absorbed into the blood, affects the brain, and is later broken down and removed. Medicines as well as herbal products also follow the same journey. In summary, pharmacokinetics helps us understand what happens inside the body, and not just drugs, but for many things we consume daily. Thank you for listening to this brief introduction to pharmacokinetics. I hope it has given our audience today some understanding of PK, and feel free to reach out for a deeper discussion and collaboration. Back to you, Dr. Hidayah.
Thank you, Dr. Norzahirah, for the interesting presentation. I believe now we have a better insight on the importance of pharmacokinetics in understanding how drugs behave in the body and how this impacts their success in development. We will now be moving to question and answer session. You are welcome to submit your questions via the link provided in the YouTube channel, and I will try my best to connect your questions to our panelists. However, if time does not permit us to address all the posted questions, our speakers will respond to each of them via email. All right, let me just go through a bit. Okay, so I think we have the first question here. Let me direct this question to Dr. Ridzuan. So, Dr. Ridzuan, are we failing because the drug is bad, or because we chose the wrong biological target to begin with? Dr. Ridzuan?
So to answer these questions, most of the time, it's not the drug itself is bad. It's that the biological target may not be the right one. In early research, we choose a target that we believe drive the disease, but human biology is, is very complex. Sometimes that target turns out to play a smaller role than expected in patients. So even if the drugs hits the target perfectly, the clinical benefit may be limited. So this is why better understanding of disease biology and target validation is very important today. Over to you, Dr. Hidayah.
Okay, thank you, Dr. Ridzuan. I believe we have more questions for you. All right, so the next question. We use animal models and in silico or AI simulations, yet humans still react differently. So where is the loss in translation moment happening most frequently?
Right, thank you for the question. By Mr. Raymond Chin, use animals and in silico. Okay, I think I may right or not. So the answer might be the loss in translation moment usually happens when moving from preclinical model to humans, and animal models and AI simulations help us actually predict the outcomes. Just a prediction. It's not the actual results, because they cannot perfectly capture the complexity and genetic diversity of human biology. Of course, when we talk about genetic diversity in human biology, it actually involves lifestyle, immune response, disease variation as well. So because of this, I think the drug that works well in the lab or animals may not produce the same effect in humans. That is one of the reasons. It's because of lack of genetic diversity between the animal and human. So AI is just a prediction.
Okay. Maybe next week turn to Dr. Zahirah for the question, right? So Dr. Zahirah, is pharmacokinetics more important than pharmacodynamics in early drug development?
Right. So I think in early drug development, both PK and PD must be given consideration. But as the drug moves in the drug development pipeline, I think PK will then be the factor that either stops the drug or says that the drug can go forward or not. But to say it is more important or not, I think it depends on the what disease that we are targeting, because there's always a trade-off between PK and PD. So maybe the drug could slightly be not very good, have not very good PK properties, but the PD outweighs that, or not very good PK properties, maybe in terms of toxicity or something like that. So maybe the PD outweighs that. So we have to see all of the data to be able to decide which is whether or not the drug should move forward. So hence why evaluation of both PK and PD at the same time is something that we are looking into right now, because if we evaluate PK separately and PD separately, we cannot see the true effect together. So we need more technologies such as the sensor technology to be able to see both the effect of PK and PD at the same time. So that will give us an insight into what is actually happening when something, the efficacy is happening, what is happening to the compound concentration as well. So I couldn't say if it's more important or not, but I think it should go hand in hand.
All right. Another question for Dr. Zahirah. So, for herbal preparation, is it scientifically valid to measure only one marker compound?
Okay, for herbal preparation, is it scientifically valid to measure only one marker compound? Okay, so in herbal, as we know, it is a complex mixture of compounds, and I think it depends on the objective of the study. If you are trying to identify the herbal plant, maybe you can only identify or measure a single compound, and that could be valid for the standardization or the quality control purposes. But I think we need to characterize more compounds in the herbal medicine because it could be the compounds that are used for the standardization is not the biologically active compounds. So I think it's important to not only rely on one marker because it might not fully represent the whole in vivo behavior of that mixture. And also in the herbal mixture, it's not just the different chemical compounds, there are also proteins and maybe some carbohydrates also. So these are the things that could affect the ADME properties that I was talking about just now. So the more compounds or the more things that we can follow along their PK journey, the better, I think.
Okay. We have a line of questions, so let me pick one. All right, so Dr. Ridzuan, so is monkey model mandatory for preclinical experiments?
Yeah, I think monkey. Of course, I think that the closest to human is the monkey. Yeah, because but it is the use of monkey is very limited because you have to consider the ethical of use of the monkey as well. So actually, it's not a mandatory for clinical experiments because it depends on the regulatory agencies as well, how they actually look at the results, whether or not your result have to be include the monkey data. Some safety data, for example, they need more than one species, but it doesn't necessarily to be monkey species. It has to be non-rodent species. So actually you can choose rather than monkey, you can also use rabbits or dogs. So monkey normally usually used when the drug specifically targets the biological pathway or reaction that doesn't exist in other species, but it exists in humans. So maybe monkey is the best in that point. So as we can see that previous COVID-19 research, they use some of the vaccines that they have studied. They are testing in the monkey as well because of the they want to make sure that the similarity of the reaction in monkey and human. Most of it are used for certain biologics and monoclonal antibodies because these are very important things at that time or at the pandemic time because they want to make it very fast testing into human trials. That's why they use monkeys. So it's not necessary, it's not mandatory for you to use monkey in clinical studies. It depends on what you are looking at. Or over to you, Dr. Hidayah.
Yeah. So can compounds in a mixture improve each other's pharmacokinetics?
can compounds in a mixture improve each other's pharmacokinetics? Yes, I think it is a possibility that within the complex herbal mixture, it can some of the compounds or something that's present in the mixture can influence or also improve each other's pharmacokinetics. But also, it could be the other way around. It could also make the PK of certain compounds less or something like that. So this happens because maybe some of the compounds inhibit the metabolic enzymes, and then that then allows a second compound to pass through better. And maybe some components may improve the solubility of the compounds, of the other compounds. And also maybe there are changes that is caused by the compounds to the gut permeability as well. So there could be improvement, but we also must be careful if there could also be some the compound is less effective than we want it to be. Yeah, thank you.
Back to Dr. Ridzuan. So how can organ-on-chips technology be used to replace the animal models?
Well, I think yes, organ-on-chip is very famous now, but you have to bear in mind that the organ-on-chip is still under development. What I can say whether or not it can replace animal or not is still in questions, because at the moment most of the published articles are they use the organ-on-chip data as a supplement. Because there's a lot of things that needs to be done in order for the organ-on-chips to actually mimics the complex biochemical process of in human. So the answer is yes, it can in future, but not now. And then you have to bear in mind that the use of organ-on-chip has to be regulated by the regulatory authorities. At the moment, only FDA has actually released the Modernization Act 2.0, which actually give a free, what we call it, make the researchers now to look at the new technology such as organ-on-chips to replace animal in future. Over to you, Dr. Hidayah.
Okay. Next question. Dr. Zahirah, how could the new drugs development target the process in ADME? What would be the best way to approach this?
So as I introduced just now, there are 4 main processes: absorption, distribution, metabolism, and excretion. So for a new drug, it has to address all these 4 to be able to be present in the body long enough for it to have an effect on the target site. So for absorption and also for metabolism, for example, we can design a drug that is a prodrug. So meaning when we take it, it is in an inactive form. And then due to the processes of the liver, when it is metabolized, it becomes activated after it passes through the liver. So prodrug is also one of the approaches that we can take as well. And for example, if we found that our new drug is broken down too quickly by the enzymes in the liver, for example, the CYP enzymes, then we can sort of block these metabolic sites using, for example, another compound, or to co-administer with our drug. So that is also one approach for the metabolism as well. And designing of the drug and changing the functional groups are also important to sort of modify the drug so that it will not be it will not fail in later stages. Yeah, I hope that answers your question. Back to you, Dr. Hidayah.
Thank you, Dr. Zahira. My next question. My next question is to Dr. Ridzuan. Historically, drugs fail due to toxicity. Today, they often fail because they simply don't work as well as expected, or in other words, lack of efficacy. So why has the needle shifted, Dr. Ridzuan?
Okay. As we know, many drugs fail because of toxicity. Maybe because the safety testing tools is actually limited. As we all know, our clinical toxicity studies, screening technologies, and regulations are much more stronger. Is there any unsafe compounds are usually detected earlier? Can be detected earlier lah. So because of that, most drugs actually entering clinical trials are actually relatively safe. But the point that we have to highlight now is that although it is safe, the efficacy is actually the one that causes the problem. So back to you, Dr. Hidayah.
Okay, thank you, Dr. Ridzuan. Okay, one more question. Right, so Dr. Zahira, how are herbal waste products usually removed from the body?
After the liver metabolizes the herbal compounds into a more water-soluble metabolites, then it gets sent to the kidney, and it is eliminated in the urine. So if the compounds is not soluble and it's larger or it's fat soluble, so instead it will be secreted into the bile and the liver, and then that will then move to the large intestine and then secreted in the feces. So, but also from my experience, I think from doing PK studies, some herbs, after we analyze the drug concentration profile against time, the concentration rises and then falls, but then there is often a small peak again at the end. So we think that some herbs undergoes enterohepatic recirculation. So it happens when the gut bacteria reactivate the waste product and then allows them to be reabsorbed into the bloodstream. So then we can often see a little peak at the end. So this is also another way that the herbal compounds get recirculated, and also then it will be then removed as well. I hope that answers your question.
Thank you, Dr. Zahira. One more question. I think we have a few minutes more. Right, so back to Dr. Ridzuan. There is a high failure rate when attempting to translate preclinical findings into clinical trials globally. So what is the local measure to overcome these challenges?
Thank you for the questions. I think, as I mentioned just now, most of the reason the high failure rate when attempting to translate clinical findings into clinical trials is because of the lack of robust data. For example, we have to actually look in every aspect of the studies. For example, we need to look at the robustness of the pharmacokinetics data. This is where you can actually look for the best drugs that is bioavailable in your body. Yeah. And then next is the pharmacodynamics data. Pharmacodynamics is the one that is very important as well, to look at the mechanism of actions and also the effectiveness of the compounds or drugs in the body. Again, we also need to conduct a proper toxicology studies based on the design of the treatment, and also the duration of the treatment, because in the toxicology there's a lot of experiment that needs to be done For example, general toxicology that consists of acute toxicity and also repeated dose toxicity and also chronic toxicity depending on the disease and depending on the treatments. And we also have other studies as well, such as reproductive toxicity and also mutagenesis study. All this actually data has to be integrated, and that has to be what you call it, has to be make it available to the regulators. If the data is properly actually analyzed and analyzed and also collected and reported, so I'm sure this will actually reduce the failure rate of clinical trials. I hope I have answered your questions. Back to you, Dr. Hidayah.
Thank you, Dr. Ridzuan. We still have a few more minutes. Right, let's go for the next question. Dr. Norzahirah. So can we design a drug with ideal ADME, or is it always a trade-off?
Right, so I think we aim to design drugs with the ideal ADME properties. This is our guidance to have a good absorption, distributed to the target site that we want, good metabolic stability, and also that we can predict when the drug will leave the body. But in reality, I think there's always a trade-off. For example, if we increase the lipophilicity of the drug, that improves the permeability and the absorption. But then that also causes the tissue accumulation and also reduced clearance of the drug. So improving the lipophilicity, will also have some trade-off there as well. And if we prolong the metabolic stability and if we design it to be more stable in the liver, it will also possibly cause toxicity as well. So that's why we need all these robust tests in order to truly design a drug that is effective to the target that we want. But I think this is our guidance. But in reality, I think there's always a trade-off. So it's important to discuss within the team as well, from the drug discovery and also drug development processes. So it's actually about balancing the development, the drug, and what happens in the body as well. So ideal drug is, I think, maybe it does not exist. I think all drugs would have side effects as well. So these are the things that researchers have to think about when we design new drugs. Thank you very much. Over back to you, to Dr. Hidayah.
Thank you, Dr. Norzahirah. So in the interest of time, we have to end our webinar session for today. As mentioned, all other questions will be answered by our diligent speakers via email. We hope this webinar has shed light on some research work that we are conducting in IMR and would spark interest as well as initiating research collaboration with others. As we approach the end of this webinar, I would like to thank both speakers for their utmost effort for this webinar session and sincerest appreciation to all the participants for your time and support to make this session a successful one. Don't forget to subscribe to our National Institute of Health Malaysia YouTube channel. And before I conclude, I would like to humbly request everyone to mark your calendars as well our next session of IMR-SEAMEO Webinar Series session with the theme Optimizing Process Flow and Resource Allocation to Reduce Wait Times at Emergency Department in a Tertiary Public Hospital, a simulation-based study that will fall on 24th of April 2026. So do join us. And thank you again from all of us in IMR. Until then, let's move forward and have a good day. Assalamualaikum.